Hi, BISC 10 students. Today we're going to go over Chapter 18, Nervous System Infections. This chapter is condensed. I'm not covering every single organism that's listed in your book. I'm going over the ones that I feel are most important or most interesting, and those are the ones you should focus on. All right, so let's get started with some general information regarding the nervous system. So some important vocab that we need to know are the meninges and what they are. Those are three specialized layers that encase the central nervous system. And if you don't remember what the central nervous system entails, that is the brain and the spinal cord. So you have these three specialized layers that surround the brain and the spinal cord called the meninges. Also surrounding the brain and the spinal cord is a fluid called CSF, the cerebrospinal fluid. It's this kind of viscous fluid, thickish, that cushions as well as nourishes the central nervous system, your brain and your spinal cord. And then a concussion is injury to the brain and or spinal cord caused by physical force. Now, we're not going to focus too much on concussions here. I may bring it up again for a moment, but just giving you some key terms, some important clinical vocab. We'll see a few more bits of clinical vocab that are important. So right here, inflammation, you should remember this from earlier chapters. Remember I mentioned anything that ends in itis is referring to inflammation. So what is inflammation? Basically, we're moving plasma and white blood cells to the damaged or infected tissue. It's a body's natural response. So for instance, if you ever have had a cut on your arm, for instance, and it feels warm to the touch. So there's a clot that's formed, it's not bleeding anymore. You touch it after, let's say, a couple hours up to a day later, and it feels warm to the touch, it's a little bit raised. That's because it's what we call site-specific inflammation. The body is sending white blood cells and plasma to that area to help fight off a potential infection. Meningitis is inflammation of the meninges, so the three layers that surround the brain and or the spinal cord. Encephalitis is specifically inflammation of the brain, and then meningoencephalitis is inflammation of both the brain and the meninges. So it's important to know these terms because I will be using them throughout the entire chapter. As I said, the central nervous system contains both the brain and the spinal cord. Everything else off of that is considered the peripheral nervous system. Now, it doesn't mean the peripheral nervous system isn't important, because it is. It's very, very important, especially with transmission of diseases. Diseases that affect the nervous system, specifically the central nervous system, they don't necessarily start there. They enter the body through the peripheral nervous system and travel to the central nervous system. So the peripheral nervous system, which is everywhere other than the brain and the spinal cord, is just as important as the central nervous system. Now, in functionality, yeah, the brain's a bit more important and the spinal cord's a bit more important than being able to feel touch, temperature, or pain in your hand. But in terms of disease, the peripheral nervous system provides a pathway to the central nervous system. So this image is a great image. I really like this one. It shows the three layers of the meninges that are around the brain and the spinal cord. You have, from outside to inside, you have the dura mater, which is the outermost, the arachnoid matter, which is the middle. Then, if you're looking inside the cranium, you have what we call the subarachnoid space. You have this space right here, which has some of that CSF in it. And then the innermost layer that's flush against the brain is the pia mater. So out to in, dura, arachnoid, pia. And if you look at this cross section of the spinal column, it's the same thing. The dura mater is the outermost. The arachnoid is next. You then have your subarachnoid space, followed by your pia mater. You need to know the order in which they are lined up, and we'll talk more about these later on when we get into meningitis. So the meninges have several functions. Some of the important ones are listed here. Supplying nutrients to the central nervous system, the brain and the spinal cord. Removing waste, so cells make waste as part of their metabolic functions, so they remove waste. And then you can it acts as a protective barrier. So what's interesting is your brain inside your cranium, it's sitting in there and it's surrounded by not only the CSF fluid, right, the cerebrospinal fluid, but also the meninges. And the brain can actually shift. Let's say you are running and you fall. The brain, as your head goes forward, and let's say you hit it on the ground, the brain kind of moves forward towards the front of your cranium. Now, because of the CSF and because of the meninges, most of the time the brain won't actually physically hit the bone, right? But in some cases, if it's bad enough, the contact is hard enough, the brain will actually make contact with the cranium and cause damage to the brain itself. But there are protective layers like the meninges and the cerebrospinal fluid to help soften the blow and actually to prevent physical damage from a hit to the head in whatever manner that may occur. So meninges and CSF are very, very important. CSF produced by structures within the brain. In that subarachnoid space, you have CSF, and it acts like a cushion. So the brain kind of like sloshes around inside the CSF, allowing it to move around without doing damage to the brain itself. Another layer of protection is the blood-brain barrier. I may refer to it as the BBB. So the blood-brain barrier delivers nutrients and oxygen to the central nervous system. Very few molecules can pass from the blood vessels that make up the blood-brain barrier into the central nervous system. And the molecules that can are lipid-based and or very, very small. Now, the blood-brain barrier is made up of the smallest blood vessels that we have in our body called capillaries. Capillaries are only one cell layer thick, so it allows, in the body, it allows for easy passage of molecules to move in and out of the capillary itself within the capillaries found in our body. We find lots of them in our lungs that surround the alveoli, right, the air sacs, to allow for gas exchange and waste exchange. Whereas in the brain, it's a bit more picky. They have what we call tight junctions, which are these junctions where the cells are kind of stitched together to the point where things can't pass through them. So the capillaries associated with the blood-brain barrier are very limited in what they allow to pass through them into the brain. And this is very important because it protects our brain from sometimes specific pathogens, right? Bacteria. It'll protect from toxins, anything that could damage the brain by limiting the ability of those substances or organisms to get from the capillary into the CSF or into the central nervous system. Bacteria require the ones that can cause meningitis and can get through the blood-brain barrier have to have a very specific set of virulence factors. So there's not a ton of organisms that can actually do this, and the ones that can have very specific traits. Viruses, since they're smaller, they do have a little bit of an easier time entering the central nervous system, but it's still not easy. And then the nervous system infections basically are more common when you have some type of injury to the central nervous system, like a concussion, or if there is an issue with your immune system. Um, we've kind of talked about at length how diseases of the central nervous system um, are very severe, but your immune system has to be, many cases, doesn't always have to be, but in many cases has to be kind of suppressed or depressed in some manner. Our immune system is really good at what it does. So sometimes it can let a few things bypass its protection, and it'll those organisms or pieces of organisms will cause inflammation to the meninges or the brain itself. Sorry, there you go. This is table 18.1. It's talking about meningitis versus encephalitis. Meningitis is inflammation of the meninges. Encephalitis is inflammation of the brain. We're only going to really talk about meningitis. We're not really going to focus on encephalitis. You can see that there are some common causative agents here. Strep pneumoniae, we'll talk about that organism. We'll talk about Neisseria meningitidis and Haemophilus influenza. We'll talk about these bacterial pathogens that will cause meningitis. Now, viral meningitis is more common than bacterial meningitis, and bacterial meningitis is typically much more severe than viral meningitis. There are some really common signs and symptoms associated with meningitis that you should definitely know, because all the organisms we talk about will have some of these same signs and symptoms. So, fever, headache, stiff neck, nausea, vomiting, and even photophobia, so light sensitivity. These are things that are very important and to look for. If a patient, let's say, gets cut with a piece of glass, or comes in contact with somebody else who has strep pneumoniae or Haemophilus influenza, it's possible that if they get it, some of these initial signs and symptoms are really what you need to be paying attention for, especially nausea, vomiting, stiff neck, and photophobia. Those are things you should look out for. The sensitivity to light is one of those symptoms that really is just very important. How do we diagnose it? We take a sample of CSF via a lumbar puncture. Basically, we go through between two of the lumbar vertebrae, and we pull out some CSF, and then we can run some tests on it and culture it, etc. For viruses, there are no known vaccines for the ones that cause viral meningitis. But again, with antiviral treatment, viral meningitis is easier to treat and more common than bacterial. Bacterial meningitis is actually prevented by vaccines in some case. For instance, Neisseria meningitidis and strep pneumoniae and Haemophilus influenza, all three that we'll be talking about, have some form of a vaccine, and then we can treat them with antibiotics. As I mentioned, viral meningitis is more common than bacterial, but bacterial is a bit more severe. So, viral meningitis more common, bacterial less common, more dangerous. You can also have infections in the peripheral nervous system. I've mentioned that. Some viruses hide in the peripheral nervous system and cause occasional flare ups. A really common one that people know about, that they're familiar with, is HHV1, human herpes virus 1, which causes cold sores. When a person is infected with this virus, instead of the virus being in the central nervous system, it actually hides in the peripheral nervous system. And it hides there for however long. There's really no good way to figure out how long it's going to hide. It becomes a latent, what we call a latent infection, and we talked about latent infections earlier in the semester. So that's why people who have this infection, human herpes virus 1, which causes cold sores, they don't have cold sores all the time. They randomly pop up, and that's because they hide until they decide to express themselves. And when they're expressed, that's when you start to see the symptoms, and you see the actual sores themselves. And then there are some toxins that can be made that can disable the peripheral nervous system. We'll talk about tetanus and botulism later on, and we'll talk about how they make very specific toxins that deal with the neurotransmitters of the peripheral nervous system. Some more vocab. Cytolytic. Basically, this is an organism or compound that causes lysis or rupturing of a cell. We know what a zoonotic disease is, a disease that goes from animals to humans. Systemic infections, we actually talked about in an earlier chapter. This is an infection that affects the whole body and it travels throughout the blood. And paresthesia is something you may have actually experienced before. It's a neurological symptom that is kind of like this pins and needles sensation. If your foot has quote unquote gone to sleep and you feel that pins and needles sensation, the paresthesia, that's pretty much what this neurological symptom is. And some pathogens can do damage to the nerves in the peripheral or the central nervous system that leads to permanent paresthesia, permanent pins and needles sensations. And we'll talk about that a little more later. So the first one we're going to talk about is rabies. Rabies is an interesting virus. It's part of the Rhabdoviridae family, and it is has a single-stranded RNA genome. So its DNA is its genetic material, sorry, not DNA, its genetic material is single-stranded RNA. It's enveloped, meaning it has an envelope surrounding the genetic material, the genome, the RNA genome. And who can be affected? Well, anybody really could potentially be infected if they come in contact with an animal that is infected. So really anyone is at risk if they come in contact with an organism that carries the virus. Now in the US, there's very few cases per year. We're talking less than 10 per year in the US. However, in other countries, this is actually a lot more common, and typically it's usually mostly Africa and Asia where this virus is most deadly, and usually about 50,000 deaths per year in that region. Um, when it comes to the symptoms, the signs and symptoms, the initial ones, uh, common to what we talked about with meningitis, fever, headache, um, paresthesia, and then disorientation, and then another one that's kind of common, or that's kind of common to this pathogen, is hydrophobia, which is fear of water. Now, it is not a literal sense of they see water and they run away. That's not what hydrophobia is really referring to. Um, this hydrophobia is the fact that the ability to swallow has become almost impossible. So when they are foaming at the mouth and they can't consume any water, it's because of the fact that they're unable to swallow. Um, the back of the throat tends to close quite a bit, which causes this foaming at the mouth. That's why if you see animals that are foaming at the mouth, that typically indicates rabies, because of the fact they can't really swallow water, and they end up foaming at the mouth. Uh, Symptoms are typically caused by the fact that the virus enters in the muscle cells and it replicates pretty slowly. We're talking a couple weeks to a couple months before you start to see signs or symptoms. And eventually it'll travel up the peripheral nervous system and invade the central nervous system. Okay, so we will see that. So it can move from the peripheral nervous system up to the central nervous system. Now, when it comes to rabies, the pathogenesis is not really well understood, but we do know it doesn't involve cytolysis, so it doesn't actually kill the cells and destroy them, which makes sense because viruses need host cells. They can't survive on their own. So it makes sense that cytolysis is not common when it comes to the rabies infection. Transmission, it's a zoonotic infection, so it goes from animal to human, and typically it's either through a bite or by breathing aerosolized droppings. So animal droppings, and then let's say you till the soil, for instance, can lead to aerosolized droppings. In the U.S., the most common animals associated with rabies are typically bats, skunks, raccoons, foxes, depending upon where you live in the country. Um, it can determine which organism, which animal is more commonly associated with transmission of the virus. Diagnosis. So, diagnosis, it's kind of hard because it takes some time for the incubation period to kind of run its course, a couple weeks to a couple months. And even then, it's hard because viruses live inside of our own cells. So, yes, there are ways to detect the virus. One of the common ways is detecting within our CSF or within the blood antibodies against the virus after the patient has been bitten. So your body does mount an immune response and produces antibodies. And if we detect those in the blood or the CSF, then that tells us that the rabies virus is present or has been present before. The best way to really do this um, is prophylaxis, treating ahead of time. There is a rabies vaccine And there's also anti um, excuse me. There are also in addition to the vaccine, there's antibodies that can be given to the patient who's been infected to help fight the virus. So they actually give you the vaccine, and then they give you on top of that antibodies that help fight this virus. Now, this gives short-term protection in terms of giving somebody the antibodies. So if you've been bitten by, let's say, an animal, you should always assume that it could potentially have rabies. There's no way to really be 100% sure unless you take the animal and you test it, which can be done. But if you're not sure, if you've ever been bit and the animal runs away, you should always assume that there's a possibility that they have rabies, because most animals, when they see you, they want to run. When it comes to animals having rabies, they can be more aggressive because it does affect the central nervous system. Um, but there's also another way that the rabies virus affects the brain. Most people hear rabies and they think what we call furious rabies, where an animal gets aggressive. There's also another version of rabies where it causes more of an inability for the animal to really understand what's going on, and it kind of walks around like it's almost drunk. We actually call it paralytic rabies. So treatment, as I said, is the vaccine, and then after a person has been bit, they're going to give antibodies. Usually they're either human or equine antibodies that can be given to the patient to help with treatment. So as I mentioned, here you have, in this case, a bat that could potentially have rabies. Maybe it does, maybe it doesn't. You can't really tell without testing it. But furious rabies animals are highly excitable. They're the ones you see that are aggressive. Where paralytic, the animals seem really just unaware of what's going on. They almost seem like they're drunk, to be honest. Uh, I've seen both, and it's very interesting the stark difference between the two. How furious rabies, these are animals that will attack as opposed to run, and paralytic rabies, the animal seems to just kind of ignore the person until that person gets a little bit too close. Uh, Here's the pathology of rabies. Animal bites, the saliva transfers the virus from the animal to the human. The virus will then replicate at that site. Now, the incubation period could be a couple weeks to a couple months, sometimes a little longer. And then it starts to move from the peripheral nervous system to the central nervous system. And as it moves from the central nervous system, from the peripheral nervous system to the central nervous system, it ascends, and eventually it'll reach the brain and causes encephalitis. And the virus will then enter our salivary glands and other organs and colonize there. What's kind of um, interesting, but also pretty bad, is that um, there is no treatment that will consistently prevent death once symptoms appear. So once you become symptomatic, there is no sure way to treat a patient successfully. This is why if you get bit by an animal, you should always assume that they could potentially have rabies and go to the doctor. Um, if you don't, if you get bit and ignore it, it could become impossible to treat you once you become symptomatic. Now, again, you may not see symptoms for up to several months or even a little longer than that, but it doesn't mean you shouldn't go see the physician or the ER when you've been bit. This is cases of rabies in the US. So you can see, depending upon where you live, determines heavily what animal uh, is is commonly causing rabies cases in a given region. East Coast, it's mostly raccoons. Central U.S., it's mostly skunks with some overlap of foxes as well. And then Alaska is mostly foxes. Now, you can see that I have an arrow with pointing from fox to a little dot right there. That would be Oxford, Mississippi. My wife one day was walking to her car and she dropped her keys. And she went to pick her keys up, and from under the car, a fox bit her hand, and it bit between her thumb and her pointer finger, right in the kind of the meat of the hand. And it latched on, and it would not let go. She had to shake it off and hit it with her other hand to get it to release and run away. Now, was it that the fox had rabies and was more aggressive, or was it that it was just hiding in place and my wife got too close? You don't really know unless you can test the animal. So my wife, who is not a scientist, doesn't tell me about this until 8 to 10 hours later, and immediately I realized we need to go to the ER. Go to the ER here in Oxford, and the first thing they do is at the wound site, they give 5 shots of the vaccine at the bite location. So they give 5 shots, and that's the first step. After that, they continue giving more shots over the next 14 days of antibodies. So in Oxford, we don't carry them here. We actually don't have, the hospital did not have them. They had to be flown in to Oxford so she could be treated. They gave her additional shots with the antibodies against the rabies vaccine. They made her come back 3 days later for more shots of the antibodies. And then another, after 3 days, 5 days, 7, 10, and 14. So all those, she had to make 5 or 6 trips to the hospital to continually get more and more shots of these antibodies to protect against the rabies virus. We still don't know if the fox had rabies or not, but because I'm a scientist and a microbiologist, I know that if you leave rabies to incubate once you become symptomatic, there's really not much you can do. So you always want to assume the worst. Yes, it's inconvenient. Yes, it's expensive, but expensive and inconvenient versus death, right? So if you ever come in contact with an animal that you're unfamiliar with and it bites you, even domestic animals, if they bite you, you should seek out medical attention just in case. My poor wife has some pretty interesting medical stories. So as we continue to go on in this semester with diseases of other parts of the body, we'll talk about her quite a bit. Couple more terms that we need to know. Spastic paralysis basically is the inability to relax specific muscles. This is really closely associated with tetanus, and when we will talk quite a bit about tetanus in a little bit. Toxemia, this is where you have toxins in the blood which are made typically by bacterial pathogens. And then serotype. Serotype is basically a group of microbes that are related because they have similar antigens that stick off of their surface. And we use these antigens to determine what organism is present. We use quite a few tests and to determine what antigen is present. And one of those tests is called an agglutination test. And I'm not sure if I have it in this PowerPoint, but I do have it in another PowerPoint that I will post if it's not in chapter 18. But an agglutination test essentially is you take an unknown sample and you put it on a slide, a little bit of, let's say, CSF or just a culture, put it on a slide, and then you take a known antibody that you have had made in a lab or you've purchased, and you combine the unknown sample with the known antibody, and if they match, the antibodies will attach to the antigen, and they will clump together, and it'll look like little specks of garlic, or not, pepper, in the fluid. So it'll look kind of like flecks of pepper within a clear fluid. That tells you that the antibodies are actually binding to the antigen. Now, if you get no little flecks of pepperish looking structures, then that antibody is not matching against the antigen that's in the unknown sample. So it can also help you rule things out. But these antigens on the surface are really easy ways for us to use for detection and diagnosis of diseases, because specific pathogens have specific antigens. All right. Let's move on. So this is talking about the CSF system, how bacteria cause high pressure under a lumbar puncture where viruses don't. White blood cell count is extremely high for bacterial infections, and in CSF composition, you have high protein and low glucose. We'll talk about this a little later on in more details. I don't expect you to memorize this whole chart. I just want you to see that there's some differences between bacteria and viral CSF samples. And this kind of breaks it down. If you compare the CSF and blood glucose levels for viruses versus bacteria, viruses don't have as much glucose in the CSF. Which makes sense, right? Because viruses don't need glucose, right? They don't need them to survive. Whereas in a culture, if you took CSF and tried to plate it on a specific media, which there are specialized medias that you can use to plate CSF samples, if it's viral infections, you won't see growth, right? We can't grow them on a plate. So you would see no growth. If you see growth, then you're under the correct assumption that it's very well could be bacterial infection. All right, the next one is Haemophilus influenza meningitis. So basically, Haemophilus influenza is incorrectly named. They thought for a long time, scientists thought Haemophilus influenza caused the flu, but we were wrong. It causes meningitis. It can cause some other diseases as well. But who can be affected? Typically, young children between the ages of 4 months to 4 years. They're more susceptible. Initial symptoms are pretty much the same as most other meningitis infections. Uh, you have fever, headache, vomiting, photophobia, nausea, stiff neck. These are all pretty common initial symptoms. The cause of the symptoms is the fact that it has a carbohydrate capsule. The capsule is its pathogenic, is the reason it is pathogenic, is because it has a capsule. The reason it's called Haemophilus influenza, and then they add to the end of that type B, Haemophilus influenza B, is because the capsular antigen is the B antigen. So when we name antigens, we simply, in many cases, use A, B, C, D, E, F, G, H, I, J, K, all of those letters in order. So for Haemophilus influenza type B, that tells us it has the capsular antigen. It has a capsule that's made of carbohydrates, and it has an antigen, the B antigen, sticking off of the surface. So it gives us a very specific version of this organism. Same thing with streptococci. We can say that they have serotype A, B, C, D, whatever it may be, a letter, because it has a very specific antigen sticking off of its surface. Transmission is going to be through excuse me. Transmission is typically through wounds. It's through wounds, and then when I say wounds with this, I'm not talking about you cut yourself with an object. I'm saying there's breaks in there's sores in your mouth or throat, because mostly this is going to be transmitted through droplets. But if you're a healthy individual, you really don't have to worry as much. It's not as infectious, because you really have to come into contact with more with a person over a longer period of time, unless you have, for instance, a cut or sore in your mouth or nasopharyngeal region in your nose. Then it's easier for transmission. But most of the time, you're kind of going to have this transmission occurring through respiratory droplets. Diagnosis, well, we would take a CSF sample and culture it. We would also look for antibodies. We would look to see if there's glucose present, because this is bacterial. So we would look for all those things in CSF. We would also take your blood and culture that as well. Look for antibodies and look for the bacteria in the blood, because it can move throughout the body in the blood. And then treatment, well, there is a vaccine. So when you're younger, you get the Hib vaccine, HIB, Haemophilus influenza B vaccine. And then this is a conjugate vaccine. So remember what a conjugate vaccine is. It's the antigen plus a protein, right? It's an antigen plus a protein, because that protein helps stimulate more of an immune response. And if somebody has this, obviously, since it's bacterial, antibiotics work well. Now, that will depend when you catch this disease, early on or later. For instance, if somebody has it and they are asymptomatic for a month, and then all of a sudden they become extremely symptomatic, antibiotics could work, but it may take a longer treatment period in order for the antibiotics to be effective. But if you catch it early on, then obviously it's going to take less time in terms of treatment. Because of the vaccine, this organism isn't overly common anymore. Um, the number of cases has decreased drastically worldwide because of the Hib B vaccine. Now, this organism is extremely fastidious. If you recall, fastidious means it has special growth requirements. The organism doesn't really grow very well on most media. It grows really well on one specific medium, and that's chocolate agar. Now, chocolate agar, no, it's not actually chocolate. It just has a brown color. Basically, what it is it's blood agar, like you guys used in lab, and they heat the blood agar until the red blood cells rupture, and the media turns brown because of the oxygen and the hemoglobin in the red blood cells that's being released. So basically, it's blood agar that has been ruptured. All of the cells have been destroyed. So all the stuff inside the red blood cells is now in the media. And there are two factors that are required by this organism to grow on chocolate agar. They're called the X and V factors. X is hemin, 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 and the V factor is NAD. And if you recall, NAD is an electron carrier. So, very fastidious, okay, very fastidious, requires special media to grow. Can also cause pneumonia and otitis media and otitis interna, which is an inner ear infection as well. So there are several diseases this organism can cause, but the most common one is going to be meningitis. It is kind of easy to treat in terms of antibiotics if you catch it early enough. And if you have the vaccine, then your risk of contracting this is pretty low. Other characteristics, gram-negative, aerobic makes sense that if it's going to survive in your throat, it should be an aerobe because there's lots of oxygen that goes into your mouth, throat, nose. So it would make sense that it's an aerobic bacteria. And then it has X and V factors for growth. X is hemin, V is NAD. Neisseria meningitidis, also known as meningococcal meningitis. So Neisseria meningitidis. Who can be affected? Most people can be affected. This is one of those organisms that's more of a true pathogen where, yes, if you have if you're immunocompromised, or if you're younger or older, then yeah, you could be more susceptible to this. However, it is a true pathogen. It is absolutely a true pathogen. There is one group that we do want to watch for a little more carefully than others, and that's children under 2, because they can't get the vaccine for this. So yes, it's a true pathogen, but again, there are always going to be these high-risk groups that we want to be careful of, and that's young children, the elderly, people who have weakened immune systems. Let's see, initial symptoms, kind of similar to everything else associated with meningitis: fever, headache, nausea, vomiting, photophobia. And then there's one other thing that's not as common, that's really unique to this organism, and that is what we call petechial rash. And this rash is a bit different than your normal rash. Um, Normal rashes, when you press on them, they typically turn white due to the pressure. But for Neisseria meningitidis, the petechial rash that it creates, um, when you press on it, it actually stays red. And that is one of the more common symptoms and one of the easiest ways to identify this organism. If you have all the other factors, right, fever, headache, stiff neck, vomiting, nausea, and then all of a sudden you see a rash associated with it as well, that when you press on it stays red, it doesn't turn white. That is a telltale sign that it is meningococcal meningitis or caused by Neisseria meningitidis. So that is one of the symptoms that really sets it apart from all the other organisms. Diagnosis, you're going to take blood samples and CSF samples, and you can culture both of them. You should have some growth for this organism because it's bacterial. You would also have, this organism has a capsular antigen, so you would look for antibodies that would match the antigen on the capsule for this organism. So again, kind of talking about similar to what Hib-B has, it has a capsular antigen. If we look for a specific capsular antigen, we can test for it. If we would take, let's say, a CSF sample, put a little bit of the CSF on a slide, we would take a known antibody, an antibody that is known to match the Neisseria meningitidis capsular antigen. We mix the two together on the slide, and if we start to see clumping, those little flecks of pepper, if it looks like they're clumping, then we have a positive test. That means the antibodies are binding to that specific antigen, telling you that the antigen is present in the CSF sample. Remember, this type of test is called an agglutination test. An agglutination test. So that's diagnosis. So definitely keep an eye on the symptoms that are associated with meningitis. Look for a petechial rash that when pressed stays red. And then you can test using CSF and blood samples. You can plate them, you can culture them, and you can also use an antibody test, i.e. a agglutination test, to see if the antigen and the antibodies match. Treatment, you typically want to give them several antibiotics with this organism. Usually they give in a very specific order certain antibiotics. For instance, ceftriaxone. That's typically the first one they give, and then they test to see the susceptibility of penicillin to see if the bacteria is susceptible to penicillin, and then they could treat with penicillin. But there are other ways other antibiotics that you can use, like rifamycin and several others that you can use as prophylactic treatment, meaning you give it to them ahead of time as opposed to waiting for a full-on infection. Other characteristics, let's see, transmission, again, is respiratory droplets or direct contact with secretions. It does make a endotoxin that is made very rapidly, which is something you have to be very careful of, because remember, endotoxins are made when you have a gram negative cell because it has an LPS layer. This is a human disease as well. And I mentioned earlier we have a vaccine about 5 or 6 slides ago. I mentioned that we have a vaccine for Haemophilus influenza B, Neisseria meningitidis, which is meningococcal meningitis, and also pneumococcal meningitis. But the vaccine that we give is effective for about 5 years maximum before you need to get a booster shot. There are different versions, meaning different serotypes. So remember, this has capsular antigens, meaning it has things sticking off of the capsule. Now, depending upon which version you have, the treatment is different, as well as the fact that some of the versions like serotype B is a bit more dangerous because serotype B is almost identical in terms of structural components to fetal tissue. And the immune system can miss it. So it can actually miss this bacterial infection because in serotype B, because of the fact that it's structurally similar to fetal tissue, and your immune system is not going to attack fetal tissue, or it shouldn't. And here, this is all that information that I was just mentioning. Some people are healthy carriers. It's the way it is. Some pathogens, people are just healthy carriers. In the U.S., types B and C are the ones we typically see, even though there are vaccinations for a whole bunch of other capsular antigens, other versions, other what we call serotypes. We do recommend college students get that vaccine as well. You all live in very close, if you live on campus, close environments to one another. People who live in close environments are commonly able to transmit respiratory droplets pretty easily because of the proximity to one another. This is why college dorms are pretty dangerous in terms of infections. If one person gets sick on your floor, chances are several other are going to get sick as well because of the close proximity that you guys live in, literally right next to each other. Anyway, this pathogen is extremely dangerous, but luckily we do have a vaccine for it, and we can treat with antibiotics if there is an active infection. This is what I was talking about in terms of the petechial rash. So you can see all these rashes, all these individual rashes on this person's legs and feet. And then they start out small, typically like this or here, and they can end up being larger, almost like bruises, like this. But what makes it special is the fact when you press on it, unlike common rashes, it does not turn white. So even if you press on your fingernail, you'll notice that underneath the nail goes from red to white. That is typically what happens when you have a rash on your body. You press on it, due to the pressure, it turns white, and then when you release it, it goes back to being red. Well, in meningococcal meningitis, that petechial rash actually stays red when you put pressure on it. That is a very important sign and symptom that you can look out for. If you suspect meningitis, one of the possible causes being Neisseria meningitidis or meningococcal meningitis, you look for that rash. You absolutely look for that rash. Pneumococcal meningitis, this is caused by strep pneumoniae, which we talked about in chapter 16 when we were referring to it as a respiratory disease. But it can also cause meningitis. It can also cause meningitis. It is probably, I don't know if the statistics are still saying this, I'd have to look, but in 2018 slash 2000, early 2019, this was the leading cause of bacterial meningitis, pneumococcal meningitis, which is strep pneumoniae. And there are different age groups that are attacked by different versions of organisms that cause meningitis. So if you look here, infants and children, strep pneumoniae. Older adults, strep pneumoniae. Adolescents and young adults, strep pneumoniae. Do you see kind of a pattern here that this organism is extremely pathogenic and it can absolutely infect multiple age groups? It doesn't have to, it's not pigeonholed into one specific age group. This is a very pathogenic organism. Obviously, people who are young and old, very young and very old and immunocompromised are at a very, very significantly higher risk than somebody that's healthy, but even healthy individuals are at risk with this as it's a true pathogen. So let's talk about strep pneumoniae for a minute or two. So we know the causative agent is strep pneumoniae. We also know that the people affected will vary from the really young to the really old and immunocompromised patients to people that are healthy. It's possible. People that are healthy can actually get this. So we saw from that chart in the last slide that it depends upon, it really is just depends on the person. Symptoms, signs and symptoms, similar to all the other organisms that cause meningitis. Fever, chills, headache, stiff neck, photophobia, all the common signs and symptoms associated with meningitis. One other one that's not as common is disorientation. Some of these symptoms can lead to disorientation for certain patients when it comes to pneumococcal meningitis. The symptoms are caused by a capsule. The capsule actually allows the pathogen to adhere to the mucous membranes, so your nose and lungs and throat. Also, the capsule helps evade phagocytosis. And it also produces a toxin. So you can see why a wide range of people can be infected by this because of all these virulence factors. Transmission is, this is usually an opportunistic pathogen. It's person to person, is a little bit more rare, and person to person is a little bit more rare than respiratory droplets. So respiratory droplets would be a little bit more common, but it can go from person to person if you share bodily fluids on a regular basis. So once or twice, maybe not a big deal, but since it's a common inhabitant of your nose and nasopharyngeal region, it can be transmitted through secretions and like saliva, for instance. So that's a little less common. It's more respiratory droplets, but it is still possible with human to human transmission, but that's quite a bit less common. There is a vaccine for this, and you can get that vaccine typically over the age of 65, I believe, 60, 65 in that range, and children over 2 years old. So children under 2 years old are people that you have to be very careful with. Okay, you have to be very careful with children under 2 because they can't get that vaccine. Now, recently they've come out with a newer version of the vaccine um, that will protect children under 2 and people that are immunocompromised. But that's more of a newer vaccine that's been produced. But the original one called Pneumovax, which is really commonly given, typically given to people over 65, 60 to 65, and children older than 2. There is a new one, I think it's called Prevnar, I think. Prevnar. That one protects against several different strains of strep pneumoniae, and it can be used for children under 2 and immunocompromised patients. But that's a little bit newer than the other one, the Pneumovax, which is more, has been around for a bit longer. How do we? So with transmission, respiratory droplets, sometimes through secretions. Diagnosis, you can actually take CSF and you can try to culture it. You can try to culture blood, or you can use testing for antigens, specific antigens. Pneumococcal meningitis has a capsule, and it has a capsular antigen. Okay, so it is a capsular antigen. So again, we can use an agglutination test, or there are tests called ELISA tests, ELISA, ELISA tests. Now, I haven't really gone into what ELISA is in this class yet, but I do plan on posting a slide about what ELISA is. The name ELISA stands for enzyme-linked immunosorbent assay, and I know that literally means nothing to you guys at the moment, but it will, because I will be posting a slide on what ELISA is. Because you can use ELISA to test for many of these diseases that we're talking about in this chapter. You can use ELISA to test for different pathogens. So I will post an additional slide on Blackboard covering what ELISA is, and it'll be a very broken down version, so it won't be the most complex slide ever. I might make it kind of a simplified version of ELISA, what ELISA is and how it works. For treatment, I've already talked about the vaccines, and then you can give antibiotics if there's an infection. There are resistant strains that are starting to pop up. It's becoming an issue in terms of using certain antibiotics like penicillin. Now, it's not just resistant to penicillin, it's resistant to most of the cillins, ampicillin, amoxicillin, penicillin. So we have to use alternative antibiotics like cephalosporin, vancomycin. We use different antibiotics that target different things. So remember, penicillin attacks cell wall synthesis. So if penicillin, there's a resistance to that, let's say some patient gets a resistant strain, which is very unfortunate, obviously. They get a resistant strain to penicillin. You would want to choose an antibiotic that would attack something different than the cell wall. So you would use something like a cephalosporin or a vancomycin, which have different things that they target in the bacterial cell, in or on the bacterial cell. So, so that should cover strep pneumoniae. Other common or interesting facts about it is, or structurally, it's a encapsulated diplococcus. So I've told you that it had a capsule, but I didn't tell you that it was a diplococcus. So it's two spherical cells that are attached to each other. Di means two, remember, so diplo means two, and coccus are spherical cells. So you have two spherical cells that are attached to each other. All right, let's move on. Some other diseases associated with the nervous system: botulism and tetanus. So both of these are caused by the genus Clostridium, and the two diseases, botulism and tetanus, are two different species. I have had to get tetanus shot on 2 occasions, when I stepped on a rusty nail, and when I almost fell out of a second-story window, and I put my hands to brace myself, and my hand went through the glass in the window, and it cut across my my right wrist pretty deep. And when I went in, because the windows were old, the house was an older house, the first thing they did before they even sewed it up was give me a tetanus shot. Because remember, Clostridium, that genus, they form something very specific. They can form endospores. Remember, endospores can survive for a very long time on their own. Okay, so it's important that we try to treat for that, even if it's not there. It's still better to safely assume that it's potentially going to be present, and you get a tetanus shot. Now, tetanus shots are pretty terrible because they give you the shot at the wound site. So wherever that wound is, they literally give you a shot in the wound to prevent the organism Clostridium tetani from germinating and causing tetanus. So let's talk about botulism first, followed by tetanus. So Clostridium botulinum, who's affected? Anybody can be affected by this. In the US, there's really less than 500 cases a year of botulism, and there are several types of botulism. There's wound botulism, there's food botulism, and there's infant botulism, all of which are very dangerous. But in the United States, we see about less than 500 cases a year. And all this data that I'm giving you is backed by the CDC, and it's from the 2008, late 2017 to late 2018 year. So it's about a year and a half old, the data, so it might be a little different, but within a year and a half, you shouldn't see a significant increase in most of these diseases, as they're not really new, right? They're not novel like this coronavirus. Well, COVID-19 specifically. Coronaviruses in general are not new, but this SARS-CoV-2, which causes COVID-19, is. But botulism, Clostridium botulinum and Clostridium tetani have been around for a very, very long time. So we're used to seeing them. We know what to look for. So who can be affected? Anybody. It really depends on a couple things. Food botulism, anybody can get. If you consume food that is contaminated by this organism, you can get botulism. You can get very, very sick from this. And that's because foodborne botulism, the incubation period is less than 72 hours. So you're looking at less than 3 days. Um, and usually it's even faster than that. Usually it's 24 hours or less. But on the safe side, we say typically less than 3 days. For infants, incubation is about 3 to 5 days, and for wound is usually a little bit longer than that, about 7 to 10 days. So anybody that consumes or comes in contact with this can be affected. Signs and symptoms depends upon which version you have. Let's focus on food botulism. With food botulism, you're going to have nausea, vomiting, muscle cramps, and then eventually you'll have difficulty breathing, difficulty speaking, and you can have symptoms that are similar to a stroke. So part of, let's say, your eyelids will droop, or one side of your face will droop. And that's because of what this organism produces, which the toxin it produces. So those symptoms are associated with foodborne, and that's what we're going to focus on here is foodborne. The symptoms are caused by a toxin. So the bacteria colonizes typically in the GI tract, and then it produces a toxin called the botulinum toxin, which results in what we call flaccid paralysis, which is the exact opposite of rigid paralysis, which is tetanus. So if we're kind of focusing in on foodborne botulism, let's say somebody's canning their own food. Some people like to can their own food for later consumption. If this organism is present in the can or the mason jar or whatever they're using prior to adding the food, if it's not sterilized properly, if the container is not sterilized, what will happen is the organism will grow and it will produce the toxin as a byproduct. And what happens is when you open that food and you eat the food, you're ingesting an enormous amount of toxin, of botulinum toxin. And if you consume too much of it, it can cause all those really severe symptoms like difficulty breathing, difficulty speaking, and even the drooping of the side of the face, the eyelids and the cheeks. So the toxin doesn't, if we're talking about foodborne botulism, you don't even really need to consume the pathogen. As long as you're consuming the toxin, you'll get the same effect. Now, if we were talking about infant botulism, yes, you would have to have colonization of the organism and then have it produce the toxin. But in foodborne botulism, you have the toxins already being produced. Now, what's interesting is a lot of people aren't really aware of this, but anytime you purchase usually a glass jar of something or even some cans, the lid, when you open it, you hear it pop, and there's that little part of the lid that you can kind of press down and makes a clicking noise. So anytime you go to buy food, if that is already popped up, you do not want to purchase that product, because what that tells you is there's an organism in there producing gas as a byproduct, and it's triggered, the pressure has triggered that little safety feature to pop. And yes, it's actually a safety feature. If you buy a can of food, for instance, there's a reason why the ridge of the food is a little bit longer than where the actual end of the can is. Because what they do is they want you to set the can down, and if it sits flat on the surface, then there hasn't been any pathogen. Well, this is not always the case, but in most of the cases, there isn't any bowing of the metal can at all, and therefore there's no gas being produced inside the can, which means no pathogen. But if you have a can that if you set down and it wobbles because the bottom of the can or even the top of the can is kind of bowed out a little bit, that tells you there is a pathogen inside that can that's producing gas as a byproduct, and it is building so much pressure that it's bowing the can, and you don't want to eat that because there's tons of toxins, botulinum toxin in there, and you will get sick very quick. This is why infant botulism, couple days because there's colonization. Food botulism, you're talking less than 12 hours. If you consume a lot of it, you're talking less than 12 hours before you start to get really, really sick. Diagnosis. Basically, in the US, most of the cases of botulism are pretty mild, so they go underreported. Um, but in the US, to diagnose this, it's really important if we suspect it to diagnose it early because the toxins are extremely potent. Um, we take blood and we look for the toxins in the blood, or we can look at in the feces because remember the pathogen can grow in the GI tract if it does grow. So you would look for the toxin, and you could also run cultures on the blood as well to see if anything grows uh, in terms of diagnosis. But it's really important to see the symptoms and look at them, especially if you're starting to see a difficulty in breathing, difficulty in speaking, because what happens is it literally um, blocks the neurotransmitters from getting across the synapse to fire a muscle. This is why we call it flaccid paralysis. Things start to stop working because of the fact that the signals never make it across the synapse. To treat this, you would give somebody an antitoxin. If it's progressed significantly to the point where they're having problems breathing, they could be put on a ventilator. And then if you have if you have this, they would also give you penicillin to treat the any living cells that are in your GI tract. Um, and in some cases, they really have to be extremely aggressive in the treatment of the GI tract because it's hard to get rid of once you have it. So they may give more than one antibiotic, but penicillin is kind of the standard at the moment to treat this, as well as an antitoxin. Here we go. It's a gram-positive rod, endospore forming. It's an obligate anaerobe, meaning it does not grow in the presence of oxygen. It dies. And to prevent this, simply proper canning techniques if you can your own food. Don't leave food out for hours upon hours and then come back and just eat it. You definitely don't want to do that because it could lead to, if the pathogen is there, it could lead to having botulism. I do want to bring up with this that the toxin is extremely potent and it works very quickly. So clinical assessment is important. Getting that patient's history and understanding, okay, have they canned their own food? Did they, eat an old can of something? Have they been, to certain regions in the world? There are lots of questions that need to be asked, in order to identify whether this is botulism or not. But the easiest way to tell is by getting a sample of the blood and looking for the toxin. And then, if the toxin is found, immediately giving antitoxins to neutralize it. If you let the toxin continually work or do whatever it does, and what it does is simply blocks neural pathways, you get these flaccid paralysis problems, and that could stop a person from breathing. It could stop a person from being able to expand the diaphragm and draw in air and exhale. So it's very important that with botulism it's treated very quickly. This kind of talks a little bit about both infant and foodborne botulism. Infant, typically you don't give children, very young children, raw honey, for instance, because there's a risk of this organism being present. But the gastric acids in the GI tract trigger the fact that it creates an endospore. The endospore ends up in the small intestine or even the large intestine, and it will then germinate. Okay, it'll germinate. Once it germinates, then it's going to create lots of toxins, and that infant is going to get very sick. Foodborne, this is local paralysis of these muscles, and it's because of the fact that this toxin is triggering flaccid paralysis. What's interesting is it can cause severe constipation, which seems to be, you would think, the opposite, right? Where the rectal muscles would be wouldn't be firing, contracting, so you would actually have diarrhea. But in this case, foodborne botulism actually can cause the opposite, causes constipation. There's several reasons for that. I'm not going to go into it because it's unnecessary. Again, there's a difference between whether you're looking at infant or foodborne botulism. Okay. Also, you may know that some people use Botox. So they take a little bit of this toxin, they modify it, and they inject it into their face or other parts of their body. It can be used, sometimes it is actually medically a valid medical option, but in many cases it's used for cosmetic reasons. And people willing to inject a toxin into their face willingly, knowing that this toxin is extremely dangerous and is very potent, kind of boggles my mind a little bit. But sometimes, like I said, there is a medical need for it. Something to treat something called hyperhidrosis, as well as several other diseases. But cosmetically, giving someone Botox, the reason their muscles, the wrinkles go away is because the neural pathways are literally blocked. The muscles can't fire. The signal never goes from one muscle to the other. It is blocked, which is why the forehead stays nice and shiny and there's no wrinkles if that's where they get the shot, because the muscles can't contract. To me, that is scary. I would not want to willingly shove that in my face, inject that into my face, but to each their own, I suppose. It's just important to know that botulism is extremely dangerous and is a very potent toxin. Clostridium tetani, so tetanus. If we talk about tetanus, let's first start off with the fact that its genus is Clostridium, so it can form endospores, right? That's something to keep in mind. It's typically found in lots of places, like soil or water. But excuse me. Anybody that has an injury can be affected. This is why I mentioned when I mentioned that my hand went through a window and I stepped on a nail, that I had to get a tetanus shot because, well, the soil or rusty nails or rusted metal can hold on to these. You find these endospores. But any injury that's exposed to the environment can lead to having endospores entering into the body. And since they're obligate anaerobes, they don't just create surface infections, they go to deep tissue, deep muscle tissue, and they grow in the deeper tissue where there's little to no oxygen. And that's part of the big problem, is the fact that it grows in deep tissues. Symptoms: a rash, and then muscle spasms at the wound site. So, for instance, you step on a nail, you don't get a tetanus shot. 7 days later, 7 days later, all of a sudden you start feeling muscle spasms where you stepped on the nail. Okay, that is one of the first indications of tetanus, because it's a toxin that's triggering those muscle spasms. So the initial symptoms: rash and muscle spasms at the wound site. Now, as the organism travels, or the toxin travels, the spasms will change areas where they occur. So the symptoms are caused by a toxin called the tetanospasmin toxin, which is released from dead cells. And what it does is it blocks the relaxation pathway of muscles. So, somebody steps on a rusty nail, they all of a sudden, after 7 days, start to feel muscle spasms at that wound site. That's because the organism is colonized and is secreting tetanospasmin, the dead cells at least are, and it's blocking the relaxation pathways. So the muscles actually keep firing and they never get a chance to relax. This is one of those pathogens where if you treat it with an antibiotic, it can make the situation much worse, because when the cell dies, it will release the tetanospasmin toxin. So you would want to use a bacteriostatic antibiotic as opposed to a bactericidal antibiotic. So the symptoms are caused by the tetanospasmin toxin, and it blocks the relaxation pathway. Diagnosis: they take a culture of the wound and they look for you can endospore stain, so you can stain for endospores. You can also try and grow it on media. Either way, they will try to identify using a wound culture. Treatment: there's a vaccination, a toxoid vaccination. Basically, a toxoid vaccination is a vaccine that gives you the inactive version of the tetanospasmin toxin. So there is a vaccine for tetanus. This is why when when you step on a rusty nail, they give you the tetanus vaccine. That is because they're giving you the inactive tetanospasmin toxin to build immunity as fast as your body can, which roughly a couple weeks. You get a booster of that, you should, at least every 10 years or so. And then if you actually step on something or cut your hand with something rusty, they're not only going to give you the vaccine, but they will also give you an antitoxin, which is basically a serum that has antibodies in it that will neutralize the toxin. So they give you the antitoxin, it finds it courses through your circulatory system, it finds the toxin and it binds to it, and when it binds to the toxin, it neutralizes it. So they would give you the antitoxin. And they could give bacteriostatic antibiotics, ones that are going to inhibit growth, right, limit its ability to grow, but also prevent them from rupturing and dying and releasing more toxin. So you have to be very careful with that. You don't want to give a bactericidal antibiotic. You'd rather give a bacteriostatic antibiotic with the antitoxin. And again, this does not, when this toxin is spreading, it creates the inability to relax for a muscle. Most commonly, we call it lockjaw. It's where the muscles in your jaw begin to lock up and you can't even open your mouth because the muscles are continually firing and because of the tetanospasmin toxin. Other characteristics, it's gram-positive rod, endospore forming, obligate anaerobe, meaning it likes environments with the absence of oxygen. And then, as I mentioned, it's common in soil, especially soil with fecal waste. Honestly, fecal contamination is one of the big issues with pretty much most diseases like this. Anything in the soil, if there's fecal waste, it's pretty common in the soil. Excuse me. This shows you the progression. So here on the left, you have what the botulism toxin does, and on the right, you have the tetanospasmin toxin. So on the left, you have the normal, this upper one right here shows you the normal way the muscle functions. The neurotransmitters jump across, they trigger muscle contraction, right? If you introduce the botulinum toxin, it blocks the neurotransmitters from being released, and this muscle never contracts, which is why we call it flaccid paralysis. The muscle never gets a chance to contract. On the other side here, we have your neurotransmitters that are going to be secreted that will, if there's no toxin present, that will contract the muscles, right? So here's a relaxed muscle. These neurotransmitters are going to be released to contract the muscle. Well, what happens is there's no inhibitory neurotransmitters, which means the muscle can never relax. So we saw over here these green spheres, they triggered the muscle to relax. And if we block them, right, if we block them, no stimulatory neurotransmitter, the muscle can't contract. Well, here, if we don't allow for inhibitory neurotransmitters to be released, then the muscle can never relax. We call that rigid or spastic paralysis. The muscle is always contracting. It never gets a chance to relax. It's continually the neurotransmitters are not inhibited and they're not released, so the muscle actually never gets a chance to relax. So I believe that's the end of this chapter. I do want to talk about one thing when it comes to food botulism. Let's get back to that for a second. When it gets to food botulism, there is one other symptom. Actually, there's two other symptoms that I did not mention that I should have mentioned. I apologize. And that's double vision and dysphagia. D-Y-S-P-H-A-G-I-A. Dysphagia. D-Y-S-P-H-A-G-I-A. Dysphagia. That means trouble swallowing. That means trouble swallowing. Okay, so that's the end of this and ask questions. Okay, have a good day and stay safe.